Yeah, so good afternoon. Welcome to BIO212 review test for chapters 19 through 21. I am going to go through those 10 review questions, paying particular attention to materials that I think students have overlooked in the past. And what I'm hoping is by giving you this sort of 2 or 3 sets of answers, meaning I'll break the 10 questions up into 2 or 3 groups, what I'm hoping is you'll come now to the review more prepared to ask more in-depth questions. Anything I discuss inside this review should be a sort of launching off point for you to go in and dig a little bit deeper. I know you've already done all the reading. I'm impressed with the way you guys are asking questions inside class. Now what I want to do is make sure you guys are looking in generally the correct direction to get the vast majority of the material under your belts. Okay? So what is the mechanism that regulates blood production? How is this measured? And what hormones would you find to indicate the mechanism has been activated? Well, when we're thinking about the production of blood, obviously what we're talking about is the major population of cells that we measure, which are red blood cells. But remember, I could be talking about any of the formed elements inside of blood. Your textbook talks about how we make red blood cells. They do in passing mention that all formed elements inside your circulatory system arise from the progenitor cells. You should know progenitor cells. You should know that you have granular and agranular cells that you find inside the circulatory system. But also, we would measure the hormones that we find inside the bloodstream. But the major hormone your textbook talks about is erythropoietin. So that's what we'll talk about here. So when I'm saying what's the major production of blood, you need water for that. Okay, so we regulate the total amount of water, and we know how we regulate that. We use the RA pathway for that, okay? Or we use vasopressin to regulate the total volume, okay? When we're talking about how we make red blood cells, though, remember we want to increase the growth of those numbers of red blood cells, so we use erythropoietin, and inside bone marrow we also require the iron and vitamin B12. We make those red blood cells, they float around for, 120 days inside your circulatory system, then they get broken down inside your kidneys. I'm sorry about that. Broken down inside your liver and your spleen, okay? But remember, I also gave you another example here, where the decrease in pressure, meaning vascular resistance, oxygen carrying capacity, and therefore the atmosphere that the body finds itself in, is detected inside the kidneys. And the kidneys' response is to increase the EPO. So as long as iron and vitamin B12 are present there, well, you're going to make red blood cells, and that's how you accommodate the change in pressure. Okay? So that's really what I was thinking when I was writing this question. Do you understand that blood is made up of all these multiple parts? Notice the textbook doesn't spend a whole lot of time developing the white blood cells. They get their own chapter. That's chapter 22. But inside chapter 19, we focus on how it is we increase the number of red blood cells, or decrease them, as the case may be. Notice we didn't talk a whole lot about decreasing the numbers of them. The next question. Each component of blood has its own unique characteristics and responsibility. What is that responsibility, and how would you characterize it? Okay? So what I'm thinking here, right? What I'm actually thinking is, if we look at that 50 mil conical tube, remember I had you guys remove 50 mils of blood from my body, and we let it sit there by gravity. You have materials that are up here. You have materials that can separate by density here, and you have materials that can separate by density here, right? Do you know what's inside each of these pieces? And I know the class is all going to say, yeah, no problem. Okay, fantastic. But what responsibility does, say, albumin have here? Or what does globulin have? Oops, a G, sorry. Globulin here. Okay, we know, we think we know what all red blood cells are doing, right? They're carrying carbon dioxide and oxygen. Is there anything else red blood cells do? I mean, indirectly they become part of a clot, but is that one of their responsibilities? What else are they doing? What's their development sort of thing? Inside here, well, you would say that this is the location where maybe we have inflammatory responses being regulated, because here's where we find our white blood cells and our platelets. So that's what I want you to be thinking of when you're thinking about the components of blood. Okay, each component of blood means that inside here we've got electrolytes as well. Well, what is ion, or what are these ions doing? You know what they're doing, right? They're responsible for the state of excitability for all excitable tissues inside the body. And if you don't have enough of them, well, we don't have an excitable organism. Okay? Hopefully that makes sense. Now go back and look at that figure. Make sure you understand what's inside each piece here, and you know what their responsibilities are. Okay, next question. Let's see. Let's get rid of this. Poorly spelled. Okay, let's get rid of that. Let's get rid of that. And ask the question again. How does clotting actually work? Is a platelet plug different than clotting? Is there a difference in the mechanism of activation? Obviously, you know that this is not a simple yes or no question to be answered. What ensures that clots are resolved? And what are the signaling molecules required to form that plug, and what shuts them off? So this could take us quite a while to answer, but I want to start at the plug itself. Because remember, plugs were mainly discussed with the presence of collagen. Which means I'm thinking, do you understand what do the actual platelets do? So I'm just going to put plates, okay, short for platelets. So the collagen interaction with platelets, those platelets are then responsible, obviously, for making, or releasing, I should say, molecules, and the molecules primarily are going to be the prostaglandin, thromboxane A2, TA2, as well as serotonin, SER, okay, and I believe it's ATP, okay. And what I want you to remember here, of course, is that just because the platelet plug has been formed, we also have a series of responsibilities. Remember, there's a vascular spasm, VS for vascular spasm, okay. I should put V.S for vascular spasm. Okay, as well as the plug formation itself, okay? And what's going to happen, of course, is then this is going to allow the next part, which of course is going to be coagulation. Okay, so I'm going to put here coagulation, coag. And that coagulation is that multi-step from both the intrinsic, and extrinsic, EXT, parts. Remember, we are either going to use the actual platelet plug to continue clotting, or we'll use tissue factor to now supplement that. Remembering that without calcium, none of this happens. Okay, so calcium is important in here, but ultimately we exit the final common pathway so that we take the soluble fibrinogen, fibrinogen, and we convert it into, fibrin. F-I-B. Okay? Now, simultaneously, while this is happening, this is why it's so important to understand what ensures that these clots are resolved, is that you also have plasminogen, being converted into and activated as plasmin to, there's an S missing there, to now ensure that we actually remove or resolve that clot. Okay? That's the level of detail you need to be thinking about. What's actually happening as we move through the intrinsic and extrinsic pathway. What about these phospholipids, okay, that allows this clotting mechanism to be called a positive feedback system? And that's because as long as there's collagen present, as long as there's a need to stop blood flow out of the circulatory system, this entire system continues to work. Okay? Cool. Next. Now you've already thought about it, can you describe the heart electrically, muscularly, through autonomic regulation and cardiac output? And obviously, what I mean by this is if you drew a heart, and I don't care how you draw your heart, obviously, you know I want to draw the heart a specific way. Okay, whoops, sorry about that. Let's go in the other direction here this time. I am so breaking this right now. Thank you. To normal. Fantastic. Good. So what we're going to do is we're going to draw the heart the way I want you to think about a heart, and then we're going to break apart what it's doing. Okay? Remembering that you know where the musculature is, okay? Or you know that if we're here, we have a really thin wall, Yay, thin wall of muscular tissue, very thin wall of muscular tissue here as well. We also have a slightly thicker wall here, and we have the thickest wall of musculature here, right? Now, think about what I'm asking you to do. I'm asking you to think about the heart electrically. Okay, so if I'm you, I'm over here and I'm going, well, electrically means I need to know about the conduction system. Conduction. Right, and that conduction system has the nodes. Okay, the nodes also are going to attach to the bundle of His, His, right? And that bundle of His ultimately causes the distinction between what's happening inside the atria and what's happening inside the ventricles. When I'm saying muscularly, you understand that electrical conduction muscularly also means that by using gap junctions, we allow then for the coordinated depolarization of each of the individual cardiac myocytes, so that you actually have a lag period that allows for contraction to occur at an offset rate. As we move from, say, the SA node over here to the AV node over here, this musculature doesn't contract at the same time that the musculature over here contracts. It's off slightly. The part 3 here is, I guess before I move on to part 3, make sure when you're looking at this you understand why the sarcoplasmic reticulum is different here inside cardiac muscle cells and, say, skeletal muscle tissue. Okay, through autonomic regulation, obviously, I'm hoping you understand what I mean is, what am I doing here? Sorry about this, folks. I'm still very, very new to all of this, and I make many mistakes with it. Okay, what I'm talking about is, by the time we finished chapter 21, we show you that both vagus and cardiac accelerator nerves. So one is sympathetic, the other is parasympathetic. Remember, parasympathetic is vagus, sympathetic is cardiac accelerator nerve. Target both the SA node and the AV nodes, but that only the cardiac accelerator nerve targets the ventricle. So that means there's going to be a differential in terms of efficacy here, and you want to pay attention to that. Okay, what does that have to do with cardiac output? Well, you know fine well that if we're going to activate the vagus nerve more, we're going to slow down cardiac output. If we're going to activate the cardiac accelerator nerve more, we're going to increase cardiac output. Now what you want to do is you want to go back into the text and figure out what the volumes are that are changing. How much blood are we pushing out? Okay, this is going a little bit longer than I wanted it to. But 13 minutes is a good place to stop. So we'll stop here and we'll pick up with the next remaining 5 questions, or 6 through 10. Okay, bye.